So to start off, would you introduce yourself? Tell us a little bit about what you do and what got you into researching about allergies.
Sure. So I'm Cathryn Nagler. I'm a professor here at the University of Chicago in both the Biological Sciences Division and in the Pritzker School of Molecular Engineering. I've been studying, I'm an immunologist by training. That means I study how the body basically how the body protects itself from foreign invaders, but more recently, how the body is perceiving things that are harmless as if they are foreign invaders and attacking them. So I've been studying how our bodies tolerate things that we take in orally, usually food, since I was in graduate school, a long time ago. The interest in food allergy is relatively recent. Before that, I was studying inflammatory bowel disease. It's a disease, we used to think it was an autoimmune disease. Now we know it's a disease in which our bodies attack the bacteria that normally live in our GI tract. And when it became clear that these bacteria in the GI tract were regulating responses to themselves, I started to wonder if they could be regulating responses to food. And that's what I've been studying since I came to the University of Chicago now 15 years.
Yeah. So, I've seen a lot of, I've read a lot about your research about the, gut microbiome and butyrate.
Mm-hmm.
So can you tell us a little bit about that and, what that is?
Sure. So over the last 10 years or so, we've identified a particular, backing up, there are trillions of microbes that live normally in a healthy state in our GI tract. Not in the upper part of the GI tract, so not in the esophagus, the upper part of the small intestine, but deep in our large intestine or colon. And they perform many functions that are critical to our health. So for example, there are some vitamins that we need for blood clotting that our body can't make on its own. It's bacteria in our gut that make it for us. We can't digest dietary fiber. Our bodies can't digest it. The bacteria in our gut ferment that dietary fiber into critical products that are used by the cells of the epithelial lining to produce energy for their own function. One of these is the short-chain fatty acid butyrate. So we have noticed, our hypothesis about why food allergies and other diseases that are called non-communicable chronic diseases that are rising in parallel, like inflammatory bowel disease, autism, obesity, diabetes, all of which are rising pretty dramatically, although not quite as obviously dramatically as food allergy during the last 20 or 30 years, that we think they're all related to lifestyle changes that have depleted protective populations of bacteria from our guts and made us susceptible to these diseases because they've affected the ability of the epithelial lining to function effectively as a barrier. So when you think about it, your gut is full of trillions of different microbes. It's a single cell layered epithelium.
Yeah.
All of those things are potentially stimulatory to the immune system, foreign to the immune system. That barrier has to be so effective that the immune system normally doesn't see those contents. So it has to be maintained very carefully. When you start to deplete populations of bacteria that are part of that process, it's easy to see how that's going to move over into pathology and start to have improper immune function. So we identified a particular population of bacteria that's important for this barrier protection, and then what we're focused on now mostly is looking at the molecules that the bacteria produce and how they interact with the host. And the first one we looked at is butyrate. So we showed that it's a natural product of the bacteria, but we also worked with our colleagues in the School of Engineering to create a formulation that allows butyrate to be released in the lower part of the GI tract where it's normally produced. So you can buy sodium butyrate from Amazon this afternoon, and if you take it orally, it's not going to get past your stomach.
Yeah.
It can't reach the lower, and plus it has a terrible smell, a terrible taste. You would never want to take it orally. Our formulation gets past all those problems. microbes. It has no smell, no taste, and delivers the butyrate to where it's normally produced.
Okay.
In mouse models, we can show that formulation protects against food allergy.
Mm-hmm. And so, like, what would you need? Like, I guess to start off, what are those like lifestyle changes?
Yeah, so many that collectively at a population level are changing our responses. So biggest culprit is antibiotic use. Not only prescribed antibiotics, so many American kids get many doses of antibiotics during the first year of life, usually for ear infections, which are mostly viral. So antibiotics don't do anything for a viral infection, but do a good job in messing up your microbiome. Then there's antibiotic exposure in our food and water supply. So I always tell the story, cattle farmers for years were giving low doses of antibiotics to their cows to make them fatter. So if the cows are fatter, more meat, more profit.
Yeah.
It turns out that what they were doing, we now know from mouse studies, is creating a disorder, a metabolic syndrome in the cattle that was making them fatter. And some people have said that this low dose exposure to antibiotics is what's making Americans so obese.
Okay.
Because that's a trend that's also increased over the last 30 years. So antibiotic use is number one. Number two, not far behind, is fiber. So these bacteria that produce butyrate need fiber. Our diet, our fast food diet of the last 30 years, even from the generation before mine, has changed radically.
Yeah.
Most of our foods are processed foods. We don't have humans for their whole existence were consuming a plant-based whole food type diet. So meaning broccoli as a whole food stalk, not broccoli that's been manufactured and processed so that it has none of its fiber left.
Yeah.
And our consumption of fiber has decreased dramatically. So we've taken away the substrate that those bacteria need to produce these helpful metabolites. And then other things are cesarean births and formula feeding, changing our lives, our living conditions. So for example, instead of we acquire our microbiome from our surroundings, and previous generations lived in closer contact with the natural environment. Now we're in buildings like this where the windows are sealed. They
Yeah.
can't even be opened. So there's no exposure to the outside environment. And that's changed. So all of these things together have changed the way that we as humans communicate with the rest of our natural environment and the way we acquire these populations of bacteria in a way that's very different from previous generations.
Mm-hmm. So is that like mostly seen in allergies or obesity or
In all of these, it's common to all of these chronic non-communicable diseases.
Okay.
For food allergies, the sharp increase was driven also by the fact that when food allergies started to begin to increase, the Allergy Association, the medical allergists, decided, to advise mothers, don't consume any of the major allergens. So pregnant women should not consume peanuts. Don't give peanuts to your babies. Wait until they're 2, 3, 4 years old to give them peanuts. We now know that was exactly the wrong advice.
Yeah.
That early exposure is what helps your immune system to learn to tolerate the proteins. So that action, that medical advice, together with the changing lifestyle, propelled this skyrocketing incidence of food allergy.
That makes sense.
I think, well, ahead of the interview, we were briefly conversing, and you also mentioned how you suffer different allergies. Maybe if you can talk a little bit about that.
Yeah, so I mentioned that I was egg allergic as a child, and I outgrew it. And there's something called the allergic march. Have you ever heard of that? Where people are hardly ever allergic to one thing.
Yeah.
They're usually allergic to many things, and it usually presents as a pattern. So I mentioned that very often babies present with skin rashes, atopic dermatitis, or eczema, and then food allergies comes next with the introduction of solid food, and then asthma at school age, and then allergic rhinitis, runny nose, watery eyes, hay fever. So I've had all of those. I've done the whole march. at some point in my life. And now mostly atopic dermatitis that seems to be largely responsive to chemicals in the environment. And that's another big exposure that I think is driving food allergies as well, but it's so hard to study because we're exposed to so many different chemicals and we don't know where to begin.
Yeah, that makes sense. So what does like a day like in your work look like? So, you know, how do you go about your research?
So now mostly, so I don't work in the lab anymore. I haven't worked in the lab for quite a while. Truthfully, mostly I'm writing grants to get money to fund the research because the research funding is poor right now.
Yeah.
But also reviewing the literature, reviewing the work of the people in the lab, going to meetings, presenting my work at meetings. So mostly studying, reading, writing, and presenting my work.
So so you mentioned the mice. Can you talk a little bit more about like those experiments and then like, you know, how you bring the like experiments with the mice to humans, or you know what that would take?
So one of the things that UChicago, one of the reasons I came here is that UChicago has an exceptional facility for germ-free mice. That is, mice that are kept in a plastic bubble. They have no microbes at all.
Okay.
And they're kept that way by maintaining them under very careful conditions. That gives you a lot of power to put back a certain bacterial population into those mice and see how that changes the immune response. So we've done a lot of work with those mice. Probably the most exciting work we've done is that we wanted a model to study food allergy. And unlike other diseases, so celiac disease, some people who study celiac disease can get intestinal biopsy samples and use them to study in the lab. But biopsy is not diagnostic for food allergies, and you're certainly not going to volunteer to give a piece of your intestine for our research.
Yeah.
So when it has no medical purpose for use. So we wanted a really good animal model. So what we decided to do, and because we're interested in the microbiome, is to take fecal samples from a group of healthy infants and a group from allergic infants and put those fecal samples into mice and create food allergic mice, but that had the healthy or the food allergic microbiome. And we were able to show that the mice that got healthy or the microbiome from healthy infants were completely protected from an allergic response to food in our model, whereas the mice that got the microbiome from allergic infants were susceptible. And then, so that was proof of concept that there was something in the healthy microbiome that we could discover and use to treat food allergies. So the first step was the butyrate. We've discovered also a particular bacteria that's abundant in the healthy microbiome, and when we transfer it to mice with the cow's milk allergic microbiome, we can protect those mice from an allergic response because these bacteria produce butyrate. So this system has been really powerful. Now we're looking at other products that the bacteria produce or don't produce to get more insight into what the butyrate independent pathways are that regulate allergy.
Could you kind of like expand a little bit on like how you get like the food allergy like from like the infant feces? Like how do you kind of infect the mice with that?
So in all of the experiments, we prepare the fecal samples in a way that enriches the bacterial population, takes off the solid matter, and we gavage it. Gavage means we have a curved needle with a ball in the end, so that everything we introduce we put directly into the stomach. And then that method is sufficient to allow the whole microbiome all over the animal to colonize over time, a couple of weeks. And then the way we've done it is that we pass that microbiome from mouse to mouse over time by transferring fecal samples from one mouse to another and create a living repository. And then we can take those living repositories to set up groups of mice to do our experiments.
So
And the experiment is that we have a cow's milk protein that's purified we deliberately sensitize the mouse to that protein, meaning giving it over and over again, and then give them a challenge with that protein. So not unlike what happens when you're challenged in, it's like OIT except that we're using high instead of low doses.
So like, how long did this research take? Like years or?
Many years, yes.
Like,
So the research and the body of work I have in food allergy started in 2004. So
Wow.
20 years.
that's a long time.
Have you seen like along with like the increase in food allergies, and also I guess food allergy awareness, have you seen like an increase in like the amount of grants that are given out and the amount of research that is being done like around you?
Not the amount of grants being given out, no. The amount of, I think more, yes, certainly when I started in food allergy, there was hardly anybody studying food allergy.
Wow.
Now it's become a topic of great interest. So there are a lot of people studying different aspects of food allergy. So I think that's very promising. We don't have any really big breakthroughs that will define a treatment. The Dupixent, you probably know, blocks one of the mediators of an allergic response, and Xolair blocks IgE. Those are the two entries into the market, but those, if you took those kind of antibodies to block your immune response, you probably have to take them for your whole life. And there's a cost to doing that. You know, we think that the immune system, the type of response that's associated with allergies, downregulates an inflammatory response, the type of response that's associated with many autoimmune diseases, and the converse is also true. So if you block that whole arm of the immune response, it may promote inflammatory responses because you've taken away the counter-regulation. So I would be worried about that. Blocking IgE, I'm not so worried about because I don't think we need IgE. I don't think it has any useful purpose anymore. It may have some earlier point in evolution, but it doesn't now.
Yeah. I had a question like about an experience that I had. So during when I was going through OIT, it was kind of near the end of my treatment. I was up to like 1 peanut, and I was doing pretty well. I did like I used to updose every 2 weeks, but with the 1 peanut, I think I was at that level for 6 months, and like a few months in, I'd just gotten a COVID vaccination, and I had my daily peanut, but that time I got an allergic reaction. Do you think,
Around the time of your COVID vaccination?
Yeah.
Wow, that's kind of surprising to me because the COVID vaccination is going to promote the other arm of the immune response.
Mm-hmm.
So I think allergies are so unpredictable. What's
Yeah.
amazing to me is that, so our mice are in a completely controlled environment. We're controlling their diet. They're genetically identical.
Mm-hmm.
Every cage is in the same environment. And the response from mouse to mouse is very variable.
Huh. So
it's just a completely unpredictable response. And that's what makes it so dangerous, obviously.
Yeah. Very interesting. So do you think, with that unpredictable response, do you think there's a reason that some people will have what do you think the reason is there are different levels of allergies? I know there's some people who even I've heard stories of kids especially with bullying, they've touched cheese and then they've passed away from that. I just wanted to...
Yeah, so that's, I've had people ask me for advice, what's the safe dose of allergen that we can, say somebody who wants to have, make a certain product and they want to say this has a safe dose of allergen. And I always say there is no safe dose. We don't know what the safe dose is because people can have a severe response to almost imperceptible crumbs on the table. So I think that's really the challenge and the safety challenge for people who have severe allergies. The other problem is that you can have a severe response on one day and almost no response on another day to the same exposure,
Yeah.
as you're mentioning with your OIT.
Mhm.
So often when kids go to college, and they're not being supervised by their parents as carefully,
Mhm.
they become much more comfortable and say, well, I haven't had a response in a long time, so that must mean that now I can eat cookies that have peanuts. No, usually it doesn't.
Yeah.
So that's the big challenge. And now we have also people who've eaten a food for their whole life presenting as food allergic as adults, which didn't happen previously. So we don't because it's an unpredictable response, even in an individual who's clearly allergic, even in a mouse who's clearly allergic, and because we have all of these environmental factors impacting susceptibility, it's really complicated to understand. Another interesting aspect is a lot of people are working on how the nervous system interacts with the immune system and how the nervous system regulates the immune system, and that's a whole new area of study.
Interesting. Yeah. So, for our audience, there's a lot of, like parents or, like kids. So, do you have any like tips for people, I guess, at any age to, prevent like having allergies? Like eating like more foods growing up or anything
So
like that.
I'm a PhD scientist, not a physician, so I
Mhm.
don't offer medical advice.
Okay.
But I would say that the scientific literature supports
Yeah.
judicious use of antibiotics, not using antibiotics for viral infections, for example, and consumption of dietary fiber. Yeah, I think the data on consumption of dietary fiber is really good
Okay.
and easy intervention, including more fiber in your diet.
That makes sense.
From earlier in life, not from infancy, but from toddlerhood.
Do you know of like any correlation between like obviously in different environments that you're living in, there's a lot of different bacteria and things like the waters and things.
So
like-
the evidence.
Yeah.
I think what I know you. Sorry to interrupt
No,
you,
you're
but the evidence that people in the developing world, where there's the idea that I'm talking about the environmental exposures was originally called the hygiene hypothesis because it was associated with the cleanliness of the living conditions and exposure to clean water. Now we talk about it more in the context of the microbiome and not in terms of sanitation. But it's clear that people in environments, less industrialized environments, farm living, are protected from allergic responses because of their exposure to, especially farms. There's a lot of evidence for that.
Interesting.
Just because you're like in like a less sanitary environment, you're being exposed to more.
Yeah.
Okay. So like, do you think for someone like Ben, for example, like if he traveled to somewhere, like on the other side of the world, or if he began living on a farm, like a different environment, do you think that like his reaction to peanuts like could change over time?
Probably not at this point because his immune system has so much memory of these reactions. Would have to be done earlier in life. But our focus is to try to find out what the key molecules are so that it's not likely that people are going to make such a radical lifestyle change, right? We can't all move to a different environment. We're not going to replace our microbiomes with the microbiomes that we had 50 years ago. So we could find out the key products of the bacteria that are important for health and reintroduce those. That's our goal. And possibly give them together with OIT, for example. So we have a study with Dr. Chacho. She's giving dietary fiber together, potato starch actually, to children undergoing OIT to see if that improves their response. With the idea that giving more of the substrate that the body uses to make butyrate, instead of giving butyrate itself, could that consumption of dietary fiber during OIT help to fortify the epithelial barrier? The study hasn't read out yet, but we plan to do. She collected fecal samples from the participants over time. It's completely blinded, and we'll get a lot of information out of that.
Dr. Chacha's great.
Yeah.
That's all the questions I have. Do you guys have anything else to ask?
I guess I'm kind of curious, like, do you see anything ever about, like, kind of, like, not so much the environment you're living in, but, like, the people you're surrounded with, like, almost like a placebo effect, like, if both of your parents had, like, a peanut allergy, and so you kind of, like, or something like that, so you grew up thinking, like, peanuts are bad, but, like, eventually your body would develop a response
So
to it?
there are, I mentioned that there are people studying neurobiology, and there are people that have particularly talked about aversion reactions, and that seems to be a real physiological response. One thing we didn't mention in this discussion is that there's obviously a genetic component to susceptibility to allergy.
Yeah.
So people whose parents are food allergic are going to be much more likely to be allergic themselves or have any kinds of allergy. And that's why it was a good question you asked me if my children are allergic. Because, and I'm lucky that they're not, because that's an inherited tendency. But that doesn't, because we've had this skyrocketing increase and genetics doesn't change and people, too dramatically from one generation to the next, genetics can't explain the change that we've seen.
Yeah, a lot of, myself included, and some of my friends in the club, one of our, the hosts of our podcast, our parents have like allergies to like animals, to various animals, but then we developed these food allergies. So you think that genetics from different allergies kind of...
I think there's probably, it's clear that there's a genetic tendency to allergies generally.
Yeah.
And I mentioned that people who have that genetic tendency, like me, are more susceptible to allergic responses across the board. So my allergy to mice probably became severe because I was so intensely exposed to them when I started working in the lab.
Yeah. I don't have anything else. Is there any way that, we can support the work you do, or any thing you'd like to say to our audience about your work?
No, I'm really glad that you're doing this because I talk a lot to mothers, more to mothers of food allergic children than to food allergic young people themselves, and what they're really concerned about is bullying and jokes. So I don't know if you remember, there was a commercial during the Super Bowl that made fun of, well, actually it didn't air. There was a commercial that was scheduled to air during the Super Bowl that made fun of food allergies. And there was such a big outcry because there's such a big audience for the Super Bowl that they succeeded in getting that part of the ad removed.
Interesting. That's
good. It's really, I think this is important work that you're doing to have people understand this is, even though this is very different from what was happening 30 years ago, it's real.
Yes.
It's a serious disease, it's a life-threatening disease, and it's not something to joke about.